Well, I'd like to start by thanking the organisers for their kind invitation to discuss this topic with you. And there's probably several of you in the audience reading your programmes thinking, why is a chap who's interested in experimental portal hypertension giving a talk about ammonia addition? Well, I hope if you'll indulge me over the next 15 minutes or so, together we'll try and answer that question. Sadly, I have nothing to disclose. I'll start with this cartoon, and forgive me, there's a lot of you that will be familiar with this process, but I think it's important to set the context of really what we understand about portal hypertension so that we can then look at some of the factors that are going to be relevant when we're moving the discussion to ammonia and how they'll be influenced. So what we now understand currently is that the main driver at the onset of portal hypertension is an increase in intrahepatic resistance. And really as that progresses, there's an increasing splanchnic vasodilatation as a responsive part of the syndrome, which goes right on to lead to increasing portal pressure as liver disease gets more advanced, development of systemic collaterals. And so really the focus of interest has been over the last 10 to 15 years, certainly more, on trying to understand the process of intrahepatic resistance. And just breaking this down for you, a lot of this is actually related to structural, ultrastructural changes in the liver as cirrhosis evolves, but about 40% of this is actually related to variable factors. And this is by no means an exhaustive list of factors, but I've particularly focused on factors that I'll allude to as I go through my talk in the next few slides. And essentially, the take-home point here is that there is a disbalance between vasodilating factors, specifically nitric oxide, and those factors which are augmented in terms of vasoconstrictors, powerful vasoconstrictors such as angiotensin II and noradrenaline. Reactive oxygen species generation and inflammatory response certainly compound the process, and we know that they aggravate further portal hypertension. A classical example would be the context of alcoholic hepatitis. So, thus far, most of the therapies over the last 10 to 20 years have been focused on trying to reduce the overall splanchnic hyperemia that results. And agents such as beta blockers do this quite effectively, having also secondary effect on reducing cardiac output and thereby flow into the liver. But as our understanding of the importance of this modifiable component has increased, we've begun to realize that we need to target certainly some of these factors and the process of inflammation. And novel therapies might include, for example, modulating the gut and trying to affect factors that are driving the inflammatory process in some of these factors. And probably a very nice way of actually trying to demonstrate this is just going through this gut crosstalk. So we know, this is some data from Jane McNaughton, one of the fellows in our group, showing that patients prior to TIPS, when one punctured the portal vein, it's possible to see a gradient whereby there's a significant increase in the amount of endotoxin in the portal vein compared to the hepatic vein. Now this is not necessarily a new concept. There are several aspects of data out there. For example, data from Pepe Sook's lab looking at bacterial DNA levels, a surrogate marker for bacterial translocation, showing that this is certainly increased in cirrhosis and associated with circulatory dysfunction. Augustine Abios in the audience here showing that certainly lipopolysaccharide binding protein correlates very well with HVPG measurements of pressure. But if we then take that from the level of the gut to the liver, we can actually see the effects of the inflammatory response. This is data from Nena Sharan, one of the previous fellows in the group, again looking at livers who are compensated cirrhosis compared with those with acute and chronic liver failure. And I hope we can all see that there's a significant increased expression in TLR4, an increasing body of data accruing showing the importance of TLR signaling in the evolution of inflammatory injury as liver disease progresses. Now this is a nice study by Vijay Shah trying to put these two aspects together, modulating the gut and trying to see the effect on portal hemodynamics and inflammatory response. And just walking you through this because it's quite a busy slide, just focusing particularly on these two groups. So essentially, these were BDL mice who then had treatment with either rifaximin or vehicle. And one can see that the use of rifaximin in these animals certainly caused a significant reduction in LPS polysaccharide binding protein, but also there was a significant reduction in portal pressure. And he quite nicely went on in this study to demonstrate that the mechanisms were related to modulation of stellate cell function through a TLR4 pathway. There's also clinical correlates of this response with data in patients with alcoholic liver disease where rifaximin was shown to lower portal pressure. So question, could reduction of ammonia be a relevant factor? And I hope as we go through the next few slides, I might try and bring you around to that thought. Modulation of inflammation, as I alluded to earlier, is also important. This is a study going back some years now where we looked at patients who'd presented with alcoholic hepatitis and asked the question, if you try to abrogate inflammation with anti-TNF, could we actually modulate the vascular responses? And so essentially with a reduction in overall inflammatory response, we were able to show a very acute reduction in portal pressure within 24 hours, which persisted over the course of the next 2 to 3 weeks while that patient was in. So what potential mechanisms might have been involved to cause such a rapid drop in portal pressure, and was there an effect, for example, on the overall disbalance of vasodilatation in the liver, specifically focusing on eNOS, because there was a significant interest, certainly at UCL at the time, in this. And again, a busy cartoon, but essentially I'll walk you through this. The thought was that eNOS expression, both at message and protein levels, was adequately maintained, but there was a significant problem in eNOS activity, with a reduction in eNOS activity. A number of authors actually had published this. So this is just a cartoon looking at the generation of NO through the dimer eNOS here from arginine, and a body of literature had accrued on looking at various regulators of NOS. So Vijay Shah's group had focused on caveolin. We'd previously looked at the trafficking inducer protein nostrin, and for the purpose of this talk specifically, I'll focus in on asymmetric dimethylarginine. This is a ubiquitous product of protein catabolism, specifically of proteins with methylated arginine residues. And to walk you through this more slowly, and it's important we grasp this context for further slides that are to follow. So essentially, if one imagines NO being produced from arginine through eNOS, this entity, asymmetric dimethylarginine, is acting as an important brake actually on NOS. And if one follows that through, there's a reduction in NO, which we would anticipate would have an increase in overall intrahepatic resistance and a reduction in blood flow. ADMA is tightly regulated by an enzyme DDH1, which is particularly present in the liver and kidney. So if we have adequate amounts of DDH1, it controls the homeostasis of ADMA and thereby controls that braking effect on NOS and normally maintains a degree of vasal tone so that you can actually keep your blood flow in the organ, in this case the liver, maintained. And we were able to show that there is a clinical correlate one can look at. So in patients who had cirrhosis, we're able to look at ADMA concentrations and relate that to increasing levels of portal pressure. Now, trying to switch this gear to looking at ammonia and trying to bring that into context, and given what I've just told you. So this is data from Finn Larsen some years ago, looking at portal cable anastomotic rats and suggesting that hyperammonemia drives an acute increase in brain water first, and this is followed by a subsequent hyperemia. So this is in the acute setting. He showed that hyperammonemia correlated with increased intracranial pressure and glutamine concentrations, and indeed a number of you in the audience here have had a body of data showing that glutamine is important in the impacting on astrocyte function, specifically in relation to swelling, mitochondrial dysfunction, the so-called Trojan horse component of changing mitochondrial permeability transition, and also interfering with neurotransmission. We heard an earlier talk in relation to the impact, perhaps, say, on GABAergic tone. And an increasing body of data looking at also the effects on cyclic GMP, particularly Filippo's lab and others, looking and showing that there's a reduction overall in cyclic GMP and impact on guanylate cyclase. But interestingly, when we look at cirrhotic patients, and this is a seminal observation from Monica Guevara, where patients with advanced cirrhosis and ascites had significant increase in the resistive index, both in the middle cerebral artery and renal artery, suggesting that there was a slightly different paradigm in cirrhosis compared to looking at the acute setting of acute liver failure. So questions that follow here, that how might ammonia actually impact on vascular function in cirrhosis? So with this in mind, this is from Mike Dorenberg's group, and the study basically looking at the effect of ammonia on astrocytes, but specifically with a pre-incubation of the astrocytes with ammonia, showing that there is a significant augmented effect of an inflammatory response and cytokines that's not seen when they are looked at individually or incubated together. And in addition to this, an increased impact on NF-kappa B generation. This has been further enhanced by further data coming through from Jay Kumar here, showing that with an increased synergy of ammonia and cytokine expression, that there is an increase in TLR signaling pathways which may promote astrocyte swelling. And I take you back to the thoughts that these processes are also thought to be important in the context of liver injury and the evolution of portal hypertension. This is data from Gavin Wright and Nathan Davies, just from our group, looking at the impact on bile duct ligated rats when one adds in an additional inflammatory insult of LPS, trying to emulate the condition of acute on chronic liver failure. And we see that in these particular animals, there's an increase in brain water. And when one tries to abrogate the amount of ammonia by using the lowering agent ornithine phenylacetate, what we see is a significant reduction in INOS, a reduction in inflammatory response through NF-kappa B, and this was also associated with a significant improvement in the pre-coma and coma stages in these animals. A more recent study in the last year from this group showed that hyperammonemia also has an impact on liver injury. So they looked at a number of biochemical markers, I'm showing ALT here, but a number of biochemical markers that they were able to demonstrate which were significantly increased after 8 weeks of hyperammonemia induced in these animals. They showed a significant increase in oxidative stress in the same animals and were able to show changes in terms of apoptosis through tunnel expression and in cell cycle function both through cyclin D1 and A. They looked at differential gene expression and through microarray and were able to show particularly an increase in TLR pathway gene expression. So again, questions, can hyperammonemia have a direct impact in portal hypertension? And if so, what is the impact, for example, on NOS regulatory biology, given what I've already shown you? Now, I'm not going to steal Bala's thunder, who's going to present this in the next talk, but essentially the punchline here, we're able to look at, again, bile duct ligated rats that were treated with ornithine phenylacetate to try and lower ammonia levels, and we're able to show a significant reduction in portal pressure, which was related to significant improvement in eNOS activity, which we found to be low at the outset in these BDL animals. And this related very closely to a change in the DDH1 ADMA balance, whereby we were able to increase DDH1 expression and lower ADMA. Now you're probably thinking, well, Raj, that's all very well, but how does that correlate with anything clinical that we can relate to? So this is data from Jaz Bajaj, published last year, where he was looking at the plasma levels of ADMA in patients who actually were developing encephalopathy and was able to show an independent association with plasma ADMA compared to markers of liver dysfunction, specifically MELD. And he also went on to show patients post-TIPS who were more susceptible to encephalopathy had higher plasma ADMA levels. Again, data from our group looking at specifically ADMA DDH pathway in the brain. This is, again, the same animal model of bile duct ligation and using the same intervention to try and lower ammonia using ornithine phenylacetate. And what we were able to show that with a reduction in ammonia, there was a significant improvement in brain eNOS activity. I'm showing here the level of ADMA in the BDL animal, which one can see again is reduced following ornithine phenylacetate, leading us to hypothesize that cirrhosis in the presence of hyperammonemia and an inflammatory response promotes DDH1 dysfunction. And I could spend the next 10-20 minutes going through this, but essentially there are a number of pathways that we think are important. I refer you to the abstract 21, Gautam Mehta, who presented a few slides earlier this morning, looking at regulation of MIRS, for example, MIR30A, which may be related to this process, and we can talk at a later time on this, but associated with an increase in ADMA when this dysfunction is occurring, having an impact on eNOS activity. And we can show that in this particular model setting, again, that we were able to improve the levels of DDH1, and this is also associated with an impact on inflammatory response and an improvement of the overall clinical context. Now, there are lots of other parallels. So we've heard that these cells have a number of intermediate filaments, that they have very close correlations between astrocytes, say, for example, and stellate cells, or other fibroblastic cells like mesangial cells, the so-called stellate phenomena. So at the level of the blood-brain barrier, controlling the microenvironment, the astrocytes are thought to use their foot processes in much the same ways that stellate cells do around blood vessels as far as controlling sinusoidal tone. So can ammonia modify hepatic stellate cell function as seen in astrocytes? Now again, this work was presented this morning by Francesco Di Chiara in the abstract number 6, where showing, and I've curtailed the data here, but just showing that the higher concentrations of ammonia, we lose these lovely spindle-shaped cells from stellate cell, which are in a quiescent state, to these cells which are clearly active in their myofibroblastic transformed state, condensing these sort of 3D images. And he also had shown that there's a significant impact in their metabolic function and in their ability to proliferate. So a significant change in the overall functional status of these stellate cells. And just as a teaser here, I put up a data which is, again, quite an old study, but looking at kidney mesangial cells and the expression of alpha smooth muscle actin. So there's a lot of overlap between the phenotypes of these types of cells. And this study looked at the use of angiotensin II. Remember I said that was another important factor driving portal hypertension, and shows that in this context there was a significant increase in alpha SMAR expression. So as a teasing point, can modulation of ammonia improve renovascular dysfunction? I'm not sure that we know the answers to that question, but it's an interesting question. Now, finally, we heard from Vicente Arroyo this morning that in the context of acute on chronic liver failure, there certainly seems to be a much worse outcome in patients who have encephalopathy. And so that begs the question, is ACLF the same as cirrhosis in an advanced state, or is it actually a different phenomenon? And again, trying to draw a strong clinical data set is difficult, but I take you back to an old study from Rajiv here, where they looked at patients who had a TIPS from an acute bleed, who clearly were very inflammatory. And out of a large cohort, 4 patients did particularly badly, and these patients had severe sepsis, hyponatremia, had an inflamed state, and had a raise in intracranial pressure. And to try and elaborate a little bit more on the mechanisms of this, they looked at these patients and were able to show there was a significant increase in cerebral blood flow after TIPS. And this was related to change in nitric oxide production. And the thought process at the time was the inflammatory response was driving perhaps an increase in INOS and changing the phenomenon. But is it also possible that ACLF in this state is imparting a change in brain blood flow? And is there perhaps a contribution of ammonia? Is ammonia acting as a damp? And I think Yalda Sharifi presented a poster on this earlier in the meeting. So ladies and gentlemen, to summarize, even the translational portal hypertension chap is quite convinced ammonia is clearly much more than a neurotoxin. Hyperammonemia promotes hepatic and brain injury and possibly other organs. I speculate on the kidney. The action of ammonia appears in portal hypertension maybe through a number of different mechanisms. There may be certainly augmentation of the inflammatory response. I've suggested dysregulation of an eNOS regulator through the DDH180MA pathway, but I think there is data to also support this with caveolin. And I've suggested that there are other cell types, such as the hepatic stellate cell, which can go into an activated phenotype and a modified function because of the presence of hyperammonemia. And this is really important from the context where we're going. So therapies for hepatic encephalopathy may have much more wider reaching impact on our patients beyond the effects on encephalopathy alone, and certainly in relation to vascular dysfunction. And I'd like to close by acknowledging I've several of the people whose data I've presented here, but this is by no means a sort of finite list. There are lots of people who've gone through the group, both past and present, who've contributed to many of the data I've referred to. And finally, just to Rajiv and Nathan for the support and your friendship over the last 13, 14 years. And thank you for your attention.